Hi, and welcome to this series exploring landmark experiments in protein science based on my book of the same name, which can be purchased on Amazon, and that link is in the description. So in this video, which is part 1 of 3 dedicated to how cells make energy, I'll talk about glycolysis and the Krebs cycle, which is where molecules required to generate energy are made. The manner in which these molecules are used to actually make energy will be covered in the next video on oxidative phosphorylation. So glycolysis comes from the Greek meaning splitting of sugar, and it's a process where cells break down sugar into smaller pieces so they can be used as energy. As we'll see later, glycolysis only produces a little bit of energy when there's no oxygen available, and this energy is quickly used up. But when oxygen is available, the Krebs cycle kicks in, and the Krebs cycle is also called the tricyclic or the citric acid cycle, to produce most of the energy that cells use and generate CO2 and water as byproducts. But to really understand this process, we have to go back to fermentation. In the late 1800s, all organisms were assumed to need oxygen to survive, and yeast was the only one known to be able to ferment. But then Pasteur came across a species of bacteria that could not only ferment, but did so even better in the absence of oxygen. So Pasteur thought that maybe fermentation had something to do with the ability to survive without oxygen. And when he tested this, he found that yeast grown under aerobic conditions, that is, in the presence of oxygen, multiplied with such remarkable activity, it would not be exaggerated to say that they multiply 100 times faster with oxygen than without. However, Pasteur found that although yeast could survive better in oxygen, it couldn't ferment anymore. And so Pasteur concluded that it must be admitted that yeast, so greedy of oxygen that it removes it from atmospheric air with great activity, has no more need of it and does without when one refuses it this gas in the free state, presenting it instead in profusion in the combined form and fermentable material. If one refuses it this gas in the free state, immediately the organism appears as an agent of sugar decomposition. With each respiratory gesture of the cells, there will be molecules of sugar whose equilibrium will be destroyed by the subtraction of a part of their oxygen. A phenomenon of decomposition will follow, and hence the ferment character, which on the other hand will be missing when the organism assimilates free oxygen gas. In the early 1900s, Young and Harden were trying to figure out why yeast juice could only ferment for a short period, and they found that adding phosphate to a fermenting mixture increased fermentation. And these are the results of one of those experiments. The x-axis here is time, and the y-axis represents the amount of CO2 generated, which is a measure of fermentation. And they found that under normal conditions, fermentation reached a steady state after about 20 minutes. In comparison, there was a huge increase in fermentation when phosphate was added. Surprisingly, an additional increase could be obtained by simply adding more phosphate after the initial injection had reached a steady state. And when the authors isolated the phosphate after fermentation came to a steady state, they found that it was bound to another molecule rather than present as soluble inorganic phosphate. And they determined that this phosphate was a hexose phosphate, and so this is a sugar with 6 carbons and a phosphate attached to it. But importantly, they found that limiting fermentation by not adding glucose, which is a type of hexose sugar, resulted in the accumulation of free phosphate, but this accumulation could be delayed if glucose was once again added. So this first column specifies what was in the mixture, either yeast juice alone or with glucose, and this column is how long the solutions were incubated for, and this last column is how much free phosphate was generated. And in this first experiment, we can see that it took only 2 days for free phosphate to reach the steady state level of 0.284 grams per 25 mils of solution when there was no glucose, but it took 10 days when glucose was added. The second experiment showed a similar trend but with faster kinetics. In this case, it took only 4 days to reach steady state. And so these results suggested to them that the phosphate might be recycled by recombining with glucose, and figured that an enzyme was needed to cleave the phosphate from hexose phosphate, since boiling the fermenting mixture prevented the accumulation of free phosphate. And so these experiments led Harden and Young to propose this equation for the process of fermentation. When you add a hexose sugar and phosphate to a fermenting mixture, you get CO2, which is carbon dioxide, ethanol, which is the alcohol that we drink, and a hexose phosphate, which is the phosphorylated sugar, along with water. In equation 2, we see the reverse reaction, a dephosphorylation, where some of the hexose phosphate is broken back down to hexose and phosphate by an enzyme. The authors noted that the first of these equations does not include the fermenting complex, without which, however, the change does not occur, and it is probable that both the glucose and the phosphate form an intermediate association with this complex, which then breaks down, giving rise to the substance on the right-hand side of the equation, and at the same time regenerating the fermenting complex. And so to confirm that hexose phosphate was split into glucose and phosphate by an enzyme, Harden and Young added hexose phosphate to yeast juice that either could or couldn't ferment and determine how much free phosphate and glucose were present. So solution 1 is yeast juice by itself, and it's used as a control to see how much free phosphate is generated when nothing else is added. Solution 2 was hexose phosphate by itself in a control solution that was in yeast juice, and again to determine how much free phosphate hexose phosphate would generate without yeast juice. And solution 3 is yeast juice and hexose phosphate together. And if we look at how much free phosphate or glucose are produced in solution 3 compared to the other two solutions, we can see that they were produced significantly more when hexose phosphate was added to yeast juice, and this indicated to them that they were split from the hexose phosphate by an enzyme and suggested the name hexose phosphatase for the enzyme responsible for the cleavage of hexose phosphate into inorganic phosphate and glucose. Harden and Young later isolated hexose phosphate, which we now know as fructose 1,6-bisphosphate, which is a fructose molecule with two phosphates, and they concluded that molecules of sugar which are involved in the reaction may be decomposed into smaller groups, and that the hexose phosphate may be formed by synthesis from these. As the formation of the hexose phosphate is invariably accompanied by that of an equivalent amount of carbon dioxide and alcohol, this explanation appears probable, as it provides a source for the simultaneous production of these substances. At the same time Harden and Young were working with yeast, Batelli and Stern were studying respiration in tissues, and they came up with the idea of culturing a cell-free system of tissue samples, kind of like what the Buckners had done with yeast, to better understand respiration in tissues. So they added finely ground tissues to water, blood, sodium chloride, and other types of solutions, and energetically agitated the flasks in a water bath at constant temperature. The authors determined that red muscle extracts cultured in blood at about 37 degrees had the highest rate of respiration, and like Harden and Young, they found that boiling the extract inhibited respiration, but that culturing them in pure oxygen significantly increased respiration. Also like Harden and Young, Batelli and Stern found that adding phosphate to their extracts significantly increased respiration, and that if they filtered the extracts, neither fraction allowed respiration by themselves, but could if they were recombined. Therefore, respiration in tissues behaved much like yeast fermentation in many ways. And to make a long story short, it was eventually realized that fermentation and glycolysis, which was more related to animals at the time, were basically the same thing, which is the following. Glycogen is the form in which glucose is stored in liver and muscles for quick energy. If glucose is not available, a glucose molecule is broken off of a chain of glycogen by its phosphorylation, making glucose 1-phosphate, which is then converted to glucose 6-phosphate by an enzyme. If glucose is available, then it can directly be phosphorylated to glucose 6-phosphate by hexose kinase. Another enzyme then converts glucose 6-phosphate into a pentose sugar, and yet another adds a phosphate to it to make fructose 1,6-bisphosphate. This part of the pathway is known as the preparatory phase, and it's energy dependent as it needs 2 molecules of ATP per glucose, which are used as a source of phosphate. But in the payoff stage, fructose 1,6-bisphosphate is cleaved into 2 3-carbon molecules called glyceraldehyde 3-phosphate, and a series of enzymes sequentially converted into pyruvate, with the production of 2 molecules of ATP and 1 molecule of NADH, which is another high-energy molecule which we'll come back to in my video on oxidative phosphorylation. From here, what happens to pyruvate depends on the presence of oxygen and the organism. In its absence, lactic acid is produced in animals, whereas carbon dioxide and alcohol are produced in yeast, and this is what basically happens during fermentation. But if oxygen is available, and this is usually the case in animals, pyruvate enters the Krebs cycle to generate most of the energy required by cells. And this is why Pasteur found that yeast incubated with oxygen didn't ferment sugars, because pyruvate wasn't converted to alcohol but instead entered the Krebs cycle. And that's what I'll talk about next, the Krebs cycle. After optimizing their animal muscle extract protocol in 1910, Batelli and Stern determined that adding succinic, fumaric, malic, and citric acids to frog muscles increased respiration. Specifically, they found that adding succinic acid could completely restore respiration in tissues that had lost their ability to respire. So the authors suggested that the potential of tissues for respiration could be assessed by using succinic acid, noting a narrow parallelism between a tissue's ability to oxidize succinic acid to malic acid and its respiratory activity. And in 1920, Thunberg wanted to know if the oxidation of succinic acid to fumaric acid had anything to do with the activation of its hydrogen, which is when hydrogen breaks off of a molecule and is taken up by oxygen. This is called dehydrogenation, and it's a type of oxidation. However, methylene blue can be used as a proxy for oxygen, which is useful because it's a blue molecule that becomes colorless when hydrogen is added to it. So Thunberg filled 2 tubes with methylene blue and added muscle extracts to each of them, and then he added succinic acid to one of the tubes. He found that only the tube with succinic acid became colorless, which indicated that it had lost at least one hydrogen in the process of being converted to fumaric acid. And since he found that the activation of hydrogen was a widespread phenomenon, he came to the conclusion that all foodstuffs were hydrogen donors, and consequently, hydrogen was the main fuel of the body, stating that, "I consider the catabolism of foodstuffs to take place in a series of continuous dehydrogenations carried out by a series of dehydrogenases. When oxygen is present, the hydrogen split off by the dehydrogenases is transported to this oxygen with formation of water." This oxidative catabolism of foodstuffs is characterized not only by this series of dehydrogenations, but also with 2 other processes, viz. Addition of water and the splitting off of carbon dioxide. Through the splitting off of the carbon dioxide, the chain of carbon atoms is shortened step by step. In 1925, David Kylan reported on an experiment in which he studied a pigment in insects and worms whose existence had been put in question by many, and which produced a specific spectrum of light when in a reduced state, but which disappeared when oxidized. He named it cytochrome, cyto for cell and chrome for color, signifying merely cellular pigment. The best material for the study of the absorption spectrum of cytochrome is provided by the thoracic muscles of the honeybee. Specimens of bees frozen at -7 degrees Celsius are allowed rapidly to thaw. The head and abdomen are cut off, and by compressing the thorax laterally with the fingers, the thoracic muscles are expelled in one mass through the anterior opening of the thorax. The muscles of 2 or 3 bees, compressed between a slide and cover slip and examined with a microspectroscope, show clearly a very characteristic absorption spectrum composed of 4 bands, A, B, C, and D, which he found appeared when the sample was oxidized in the presence of oxygen. Kylan determined that cytochrome was normally found in the oxidized state in vivo, since moths displayed no spectrum when they were calm and quiet. Here he removed the scales from the thorax of a common wax moth and attached it to a slide while still alive. In specimens of males and females which began to struggle constantly, vibrating their wings in efforts to detach themselves from the slides, the bands of cytochrome gradually appeared, band A being very clear, and band B and C appeared as almost fused in one band. When these specimens ceased to move and stopped the vibration of the wings, the bands became very faint and hardly detectable. Kylan named these bands cytochrome A, B, and C, and concluded that the number and wide range of systematic distribution of the species which show this pigment clearly is so great that it may safely be concluded that cytochrome is one of the most widely distributed respiratory pigments. In 1934, Lohmann proposed that the main source of cellular energy comes from adenosine triphosphate, or ATP, a variant of the nucleotide adenosine that has 3 phosphates instead of 1, and where the 2 terminal phosphates were easily hydrolyzed. Around the same time, Lippmann proposed the idea of high-energy bonds and introduced the squiggle mark to separate them from energy-poor bonds. And to explain why ADP and ATP are high-energy molecules, I need to introduce the concept of resonance. Remember from my first video on the history of chemistry that atoms want to have 8 electrons in their outer shell, and in the case of phosphorus, it can accommodate 10 electrons. And in phosphate, this can be achieved by phosphorus and oxygen sharing electrons, but in this case, and for reasons I won't get into, the most stable structure is one where 4 electrons are shared between phosphorus and one of the oxygens. But since phosphorus is bound to 4 oxygens, it could technically share these 4 electrons equally with any of the oxygens, and in fact, that's what what it does. The molecule can be described as resonating between four symmetrical structures, and this makes it very stable. And this is what it means for a molecule to be in resonance. But when multiple phosphates are bound together, like an ATP, one oxygen in each phosphate group is shared between two phosphates, so resonance within a single phosphate is restricted by the resonance in the adjoining phosphates, which makes it less stable than if there was only one phosphate. Thermodynamic studies also found that a maximal amount of energy is liberated in a reaction converting a molecular group of particularly low stability or high energy, such as ATP, to one of particularly high stability or low energy, like ADP. So the instability of the ATP in connection with very high stability of the hydrolyzed product, inorganic phosphate and ADP, is responsible for the large liberation of free energy when this kind of phosphoric ester is hydrolyzed. So in other words, the transfer of phosphates from ATP is energetically favored because the molecules it produces, phosphate and ADP, are more stable than ATP. We saw earlier that in the absence of oxygen, pyruvate is converted to lactic acid in animals, whereas in the presence of oxygen, it enters the Krebs cycle to generate the molecules required to make most of the ATP used by cells. In the 1930s, Hans Krebs and his graduate student, William Johnson, suspected that citric acid was recycled and found that not only was there a buildup of citric acid in the presence of arsenite and malonate, which are poisons that were known to inhibit respiration, but that its oxidation was also incomplete. So when they looked for intermediate products that might have accumulated as a result of this incomplete oxidation, they found large quantities of alpha-ketogluteric acid and succinic acid in those suspensions in which citric acid was oxidized in the presence of arsenite, and they proposed that citric acid was converted to alpha-ketogluteric acid, which was converted to succinic acid, then to fumaric acid, L-malic acid, oxaloacetic acid, and finally pyruvic acid. And they concluded that if it is true that the oxidation of citric acid is a stage in the catalytic action of citric acid, then it follows that citric acid must be regenerated eventually from one of the products of oxidation. We are thus led to examine whether citric acid can be resynthesized from any of the intermediates of the citric acid breakdown. And so to look at this, they incubated pigeon muscle extracts with oxaloacetate, which is a 4-carbon molecule, in the absence of oxygen to block entry into the cycle, and found that citric acid, which is a 6-carbon molecule, was formed. And they suggested the name triose for the molecule that donates the extra 2 carbons to oxaloacetate to form citrate, leaving it open as whether triose reacts as such or as a derivative, for example as a phosphate ester, or pyruvic acid, or acetic acid. And so to summarize the Krebs cycle so far, citric acid is converted to oxaloacetic acid in a series of oxidation reactions, and then recycled back to citric acid by the addition of 2 carbons from a then unknown 3-carbon molecule called triose. In the 1940s, Lippmann's group isolated a molecule involved in the transfer of an acetyl group, which they showed contained an amino acid derivative and pantothenic acid linked to an adenylic acid derivative via phosphate bridge. They named this molecule coenzyme of acetylation, often abbreviated to coenzyme A or simply CoA. Ochoa's group then showed that coenzyme A was required in the formation of citrate with this experiment. A mixture fit for respiration produced 0.86 micromolar and 1.26 micromolar of citrate in 2 separate experiments, but when they took out the coenzyme A, practically nothing was produced. The other columns were negative controls that showed how much citrate could be produced when components known to be required were absent, like ATP or acetate, which is where the acetyl group comes from. Chantrin and Lippmann, for their part, next investigated whether pyruvate, the end product of glycolysis, could act as an acetyl donor in the formation of citric acid, and therefore be the missing triose in the citric acid cycle. And here, the authors made use of sulfanilamide as an acetate trap, which means that if acetate is liberated in a reaction, it'll irreversibly bind to sulfanilamide, which can then be purified, and the amount of bound acetate can be quantified. And as we can see, when liver and bacterial extracts along with pyruvate were incubated together, no sulfanilamide was acetylated, but there was when CoA was added to the mix. Testing an alternate hypothesis, they also tried acetyl phosphate and found that it too could provide the acetate for coenzyme A. However, the presence of both didn't increase the amount of acetylated sulfanilamide, which showed that The pyruvate-formate exchange system yielded enough acetyl-CoA enzyme complex to bring about the acetylation of sulfanilamide. And so with these results, the authors confirmed that pyruvate was the missing triose in the Krebs cycle and proposed this equation for the reaction, where pyruvate, which is really formate with an added acetyl group, transfers its acetyl group to a coenzyme A and releases formate as a byproduct. And the authors noted that the acetyl part of pyruvate becomes joined in energy-rich linkage to the CoA enzyme. And just like glycolysis, many more people and experiments were required to elucidate this complicated pathway, and I wish I could go through them all, but time won't permit this. So I will now summarize the complicated mechanism by which sugar is used to generate most of the fuel for cells, which includes both glycolysis and the Krebs cycle. As mentioned earlier, glycolysis is a quick but low energy yielding process which uses enzymatic reactions to phosphorylate 4 ADPs to ATP and generate 2 molecules of NADH at a cost of 2 ATPs. And this is what's used for energy when oxygen is not available, or when energy is required quickly, such as during high-intensity efforts, like sprinting. But when oxygen is available, the last molecule of glycolysis, pyruvate, is converted to acetyl-CoA, and this reaction generates 2 molecules of NADH per molecule of glucose, which then reacts with oxaloacetate to produce citrate, a molecule with 6 carbons. And just like in glycolysis, a series of enzymes modify this molecule by moving carbons around or by oxidation until oxaloacetate is produced, and then the molecule re-enters the cycle for another round. In total, this cycle generates 6 molecules of NADH and 2 molecules of FADH2 per molecule of glucose, which then go on to donate electrons to the electron transport chain in mitochondria, where 26 to 28 molecules of ATP are generated by a process called oxidative phosphorylation, which I will begin to talk about in the next video. And so this brings this video to an end. I hope you enjoyed it, and if you did enjoy it and would like to know more about this topic, please check out my book, Landmark Experiments in Protein Science, using the link in the description. And if you have any questions, please leave them in the comments and I'll do my best to answer them. And I hope you'll join me for the next video, which will be part 1 of 2 concerning oxidative phosphorylation.